I think it's recording now, if I'm not mistaken. I think, did you receive a notification about it?
I didn't
No.
receive a second notification, but let's see.
It says that it's recording.
Okay, perfect. If you pressed the recording button, it should be good.
Okay, good.
Welcome, everyone. We'll get started in just a couple minutes. Good morning, Dr. Zhang. Welcome.
Hi, good morning. Hi. Nice to meet you.
Nice to meet you too. I'm so happy to have you here.
Yeah, me too. Yeah, thank you.
Let's test that you're able to share your screen
Yeah, sure.
while people are coming
Let me
in.
test.
I'm making him co-host right now, so I think he will be able to present now.
I just shared. Can you guys see my screen?
Yes,
Yes,
we
we
can
can
see your
see.
screen. It's in presenter mode,
Yeah.
so we see. There we go. That's
Yeah.
perfect.
Okay, cool.
All right. Let's give 1 or 2 more minutes for folks to continue trickling in, and then we'll get started.
Okay, sounds good.
All right, Domine, is there anyone else in the waiting room? Do we have everyone?
Not at the moment.
Okay, let's go ahead and get started. We'll probably get a few more people trickling in, but we have two very interesting talks today that I'm excited about. So, in the interest of time, I would like to first, I know there are some new folks here today. So, as an introduction, I'm the lead of the AIML AWG, and I'm joined by our communications coordinator, Damian Santiago Sosa, who's helping us today. So, our first talk is from Dr. Sai Zhang, and he's going to be speaking on genomic language models and how he's used genomic language models to identify biomarkers. So, I think this talk will have a lot of relevance for us as we're thinking about how to use genomic language models in the space biology arena. So, please take it away, Dr. Zhang.
Okay. Yeah. Thank you for the introduction. Hello. Good morning, everyone. My name is Sai Zhang. I'm assistant professor at UF at the Department of Epidemiology. Today, I'm going to talk about our recent, it's not very recent, it's kind of work from last year and about a deep learning genomic language model and how we use this model to identify a gene called CCDC146 as a modifier for AI survival. So before I go to the details of my talk, I will briefly introduce research in my lab and kind of introduce myself a little bit. I did my postdoc with Michael Standard at Stanford Genetics, and then about 2 years ago, I joined UF to establish my own lab. And the research topic in my lab is about developing advanced AI and machine learning models to decode human complex diseases by integrating genetics data, genomics data, and multiomics data, as well as many non-genetic data and EHR data to help us better understand the disease biology and help us develop novel therapeutics to cure the disease. And also, we are very interested in developing AI models to predict or diagnose diseases for individuals. So today's talk is mainly about the disease called amyotrophic lateral sclerosis, ALS for short. For those you who are not familiar with ALS, so ALS is a neurodegenerative disease that weakens patients' muscle and impact their physical function. And for ALS patients, the motor neurons gradually die, and that lead to the death from the respiratory failure. And so far, people know not so much about the disease, and what is the disease mechanism, what causes the disease, and there's no cure, effective cure for the disease so far in the clinics. However, the ALS is relatively common. The lifetime risk of ALS patients is about 1/300, and it is extremely fatal disease. So the median survival time of ALS patients is between 20 to 48 months. And here are a little bit genetic background of ALS. So basically speaking, ALS is a genetic disease. About 10% of ALS is autosomal dominant. And even for sporadic ALS patients, the estimated heritability is as high as 60%. And meanwhile, the genetic architecture of ALS is extremely complex. So both common and rare variants play an important role in the ALS etiology or pathology. And so here the figure I show on the left hand side is the summary of the genetics knowledge about ALS we know, and the survival time of ALS patients is very heterogeneous. About 10 to 20 ALS patients live longer than 10 years, like Stephen Hawking, who I think who live with ALS for about 40 to 50 years. So here we are interested in this question: what is a genetic modifier of the AL survival heterogeneity? So to solve this problem, we first performed the rare variant burden test for AL survival. We are part of the Project MIND, which is the largest whole genome sequencing consortium in the world for a specific disease. Here is for ALS. And we collected about 60,715 ALS patients with their whole genome sequence data. And we also collected their very detailed clinical information. And first of all, we performed a very standard rare variant burden test. So here we have interest in the rare non-coding variants in our genome. So to calculate the gene burden, we just map the rare non-coding variant to the target genes. So here by rare variant, I mean the allele frequency less than 0.5% in the population. And then after we calculate the gene burden for each individual, we just perform a standard Cox proportional hazard model. And you can see from the figure here, there's no hit after the analysis, which means the analysis itself is kind of underpowered. And then we kind of ask, can we solve the problem using AI machine learning, right? So the idea is we want to shrink our search space from genome-wide to certain specific genomic regions in our genome. That is very important in function regarding the disease etiology. So here to solve this, we first perform functional genomic profiling of human motor neurons, we just derive human motor neuron from iPS cells. Why we study motor neuron is just because motor neuron is a key cell type for ALS. ALS is also known as motor neuron disease. So we carry out several genomic profiling, like 3D genome structure using Hi-C and ATAC-seq for chromatin accessibility, and some other chip-seq for 3 history marks annotating the enhancers promoters in our genome, and also with RNA-seq to measure the gene expression. So first of all, we run the ABC model as I show here, to map the enhancer promoter interactions within human motor neurons using the data we generated. And based on this, we have an idea, what is a regulatory map within human motor neurons. And then for each non-coding variants located within the enhancers or promoters, we want to query what is the function, what is the functional effect of individual non-coding variants. So to solve this, we developed a genomic language model, also known as a sequence-based deep learning model, to prioritize the functional non-coding variants within human motor neurons. So here is the model structure. So basically, the sequence deep learning model, as I mentioned, and the structure is a convolutional neural network. And the input of this model is our DNA sequence, right? It's a reference DNA sequence, ATCG sequence, and the output of this model is different, epigenetic features we just measured, including, so here we just use the 8-axic and 3 chipseq features annotating the enhancer promoters in our genome. And then we train the model using our data to let the model accurately predict for each genomic region whether that region has certain peaks from the genomic profiling. And so here is the prediction performance. You can see that, yeah, we named the model as neural net. We can see that neural net can accurately predict different epigenetic features solely based on the reference DNA sequence. And then we are asking what is the functional effect of non-coding variants, right? So then we use a trained model to query the functional effects of single variants. To achieve this, we just use a technique called in silico mutagenesis. The idea is pretty simple, right? So given the trained model, and we just mutate the input sequence based on that variant. For example, the variance is from A to G. We just mutate the input sequence from A to G, and then we examine the changes in the output in different epigenetic features, right? So if the changes is very large, it means there is certain regulatory function for this specific genetic variance we are querying here. And because we have 4 epigenetic features, we just took the maximum operation to derive the final variant effect defined from the model. And yeah, so probably you may also know that there's another statistical method called QTL analysis, which can also be used to associate the regulatory function of genetic variants to the like gene expression for each gene. But QTL analysis require the access to the individual level data. We need a cohort with paired RNA-seq data and their genotype data. But here we don't need individual level data. We just need a reference. The input sequence is the reference sequence, and we don't need the individual level epigenetic features. So all of these features are actually the reference data. So here we kind of demonstrate that we can use the reference data to still query the functional effect of genetic variance. And then we ask, because the model is purely sequence-based model, then we ask, is the model learning any biology underlying the regulation, gene regulation? To study this, we kind of correlate, benchmark our neural net predictions with transcription factor binding site predictions. And so here the heat map shows the result. And because of the output of neural net is continuous, is a kind of a score from 0 to 1 indicates how likely that variant is functional. And we just set different cutoffs, from 5% to 95% to define what is a functional variance. And then we use different cutoffs to see within which range this set of functional variant defined by neural net is actually enriched with a variant that disrupt the transcription factor binding site, right? So you can see as we gradually increase the cutoff, which means we have a much more stringent definition of the functional variant. This variant we identified is kind of more enriched with the transcription factor binding site disrupting variants, which means the model is actually learning certain regulatory grammar of the gene regulation. And then, with the neural net trained, and we just use the neural net to scan all of the non-coding genetic variants within our ALS whole genome sequence data. And then we just extract all of the functional non-coding variants based on our prediction, and only focus all of these functional variants to calculate the gene burden for each gene and for each individual. And then again, we run the Cox model as we just did before. And this time, surprisingly, we got several hits. This is the result. On the right-hand side shows the result after, we call it neural net boosted variant burden test. And actually, there are four rare variants identified from the analysis. And each variant can be mapped to several genes based on the ABC model prediction. And the top case is this non-coding variant, and it was mapped to four genes based on the ABC model. And these two rare variants were mapped to the single gene, and this last variant was mapped to two genes. And yeah, then next we focus on this top hit. By top hit, I mean the functional score from the neural net is the largest one across all of these hits. So then we next we start to experimentally validate this non-coding variant in cell models because so far all of this the results are based on the computational modeling, based on the machine learning predictions. We don't know whether or not the signals are really real, right? It's probably it's just a false positive result. So we collaborate with Dr. Jonathan Kubanoff from Sheffield, and we just actually knock in this specific genetic variant we identified into human motor neurons, and then we just examine, the expression level of the four genes we predicted to be the target of this specific genetic variants. And interestingly, you can see that there is only one gene, which is CCDC146, that whose expression level was really changed. Actually, is increased after the CRISPR editing, which means that the other three gene targets are probably the false positive result, and this CCDC146 is the true target of this specific non-coding variants. And yeah, this non-coding variance based on our data is associated with over 50% reduction in the ALS survival. And this is the survival curve. And so here is the story, right? So yeah, by the way, we also did a lot of a phenotypic examination to see whether or not after knocking this specific variance, we can see certain pathology, ALS is related pathology in the cells, and yeah, it is actually true. But I just skip this result in this talk, and yeah, you can. It's this kind of a story, right? So this variance is associated with the reduction of ALS survival, and this variance is also associated with the increased expression of CCDC146, right? Which means that if we can suppress the expression of the CCDC146, we can somehow prolong the survival of ALS patients, right? This is a story, right? This is a kind of implication from our data. And next, yeah, this also indicate a new treatment for ALS. If we can design, a molecule that can suppress of the CCD146, we can somehow save the ALS patients. And to test this, we collaborate with Dr. Justin Ishida from USC, and we designed a antisense oligonucleotide ASO to suppress the CCD146, right? So to see whether or not it can, rescue certain ALS phenotypes. So first of all, we tested the CCD146 ASO in neurons, and yeah, and you can this figure, the box plot shows after the CCD146 ASO treatment, the expression level of CCD146 was indeed decreased, which indicate it is effective ASO design. And then we test this ASO in several different ALS patients' cell lines, including for sure the healthy control, and we also test in different cell lines with different ALS genetic background because the genetics of ALS is super complex. We want to test the ASO in different genetic background, including 4C9 or 72 cell lines and 2 TRDBP cell line and 5 sporadic ALS cell lines. And surprisingly, you can see from the figure in the middle, after the CCDC146 ASO treatment, all of the survival defect were rescued in different genetic background when compared with the control ASO. This is kind of indicate that the CCDC146 ASO can be broadly efficacious in respective of the genetic background of ALS. And probably you know that so nowadays so far the most effective treatment is tofersen, which is a FDA approved ASO targeting a gene called SOD1, which is also another very famous ALS gene. But this treatment can only be useful for patients with this SOD1 mutation, which only account for 1 to 2% within all of the ALS patients. But here, our data kind of show that our ASO is kind of broadly effective. And so which is probably a very promising treatment for ALS. And then this is, the cell line validation, and we next, want to test it in animal models. So here we just use a TDP-43 mouse model, which is a very aggressive mouse model for ALS, and we tested the CCDC146 also in 32 mice, and another the control ASO in about 23 mice. And the figure, this kind of figure shows the comparison between the CCDC146 ASO and control ASO in terms of different ALS phenotypes. You can see that after the treatment, all of these phenotypes in the mice were rescued, by the CCD146 ASO. And here is a survival time, a survival curve for our experiments. And you can see, the survival of those mice was, significantly rescued by the CCD146 ASO. Justin just told me that, this survival curve is the best survival curve he never seen in his lab. So it's a very promising treatment. Yeah, probably the next question you want to ask, whether or not this ASO is safe, right? So it's the kind of most important question in gene therapy. And so the current data so far we have kind of showed that the knockdown of CCD756 is pretty well tolerated in the human population. And a lot of literature showed that, the loss of function of this specific protein is kind of not so toxic. And it leads to, the male infertility based on both, human and mouse model, mouse data, but no other, data shows the toxicity of the knockdown of this gene. But right now, we are still working on proving the safety of CCD756 in our ongoing projects. So yeah, so as a summary, so in this, in this project, we designed a genomic language model to characterize the functional effects of non-coding variants, and we kind of show that how we can use AI machine learning model and to analyze, large amount of human disease data, and combined with wet lab experiments, we can somehow provide novel insight into the disease biology, and also as well as new treatment that is promising for cure the disease. And yeah, so focus on this genomic language model. So the question I think we are pretty interested in our ongoing project, and also I think is very related to the NASA AWG data analysis, is that can we use this model to study the dynamics of genetic regulation? So by genetic regulation, I mean how individual genetic variants regulate the gene expression. And we all know that this genetic regulation is not kind of fixed. It's kind of dynamic in response to different environmental triggers and lifestyles and environmental exposures. So how we can study this question, right? So the way we study this question is still to use genomic language models, because somehow it's really difficult to to do a cohort level study. For example, if we want to study how this space flight can change the genetic regulation, we cannot send 100 people to the to the space and then measure their expression level, measure their genotype. It's kind of not so achievable for now, but because genomic language models doesn't require the access to the individual level data, so we just need the reference data, before and after specified. We can still use the model to query what is the change in the genetic regulation, in response to the specified. So this is a way we want to study this question, and also we are also interested in other questions. So what is the dynamics of genetic regulation in response to fiber diet, in response to exercise, and even in response to aging. So that's a kind of a project we are working on in the lab right now. And yeah, so I think this is a great opportunity for us, to study the genetic regulation for spaceflight using the AWG data. And yeah, so at last, I want to thank all of the trainings in my lab and also all of the collaborators in the ARS project. And yeah, thank you. I'm happy to take any questions.
Thank you so much. That was very inspiring. And I have a ton of questions. So I'll just start out with one. And if anybody else has a question, please go ahead and raise your hand. So, my first question is, can you talk a little bit more about how the genomic language model outperformed the traditional variant analysis that you tried to do at the beginning, and you showed that it was underpowered? So, just kind of technically or mathematically, why did the genomic language model outperform that analysis?
Yeah, so I think that's a very good question. So I think the biggest challenging in the genomics or epigenomics field is that is a dynamic is kind of a diversity of a variant effect, right? So the variant effect is not kind of it's not one, it's not fixed across. It kind of changes in response to different cell types, to different conditions of disease or healthy, and also the factors I mentioned here, different environmental factors. So somehow it's very difficult to actually measure all of this. So first, it's dynamic. It's not easy to use a single to query all of the functions across different conditions. The second is very difficult to measure such signal across different conditions, right? So I think the power of this genomic language model is that it can unify all of the information into a single framework. And after we train the model using reference data, which is pretty accessible nowadays, and we can still query the functional effect very high resolution, in different cell types, in different conditions. So yes, and also because it's deep, genomic language model is kind of deep learning model. It can capture the long range interactions between the bases between between the sequences. So the example I show here, the input sequence is 2000 base pair, which I would say is not so long. Right now we are using Informer and Borzoi models to study the same question, and the input sequence is about from 200 KB long to 1 MB. So it's really long range input sequence to all of these models. So it can output much better prediction performance by capturing the long range interactions. Yeah,
Excellent.
yeah,
That's really exciting.
yeah. Thank you.
Um, I guess my final question, since I don't see any hands up, would be you talked a little bit, at the end about how to use this technology for space biology. So, um, how would you think about designing a spaceflight experiment, like using genomic language models, keeping in mind, I think we talked on email about this, most of our data is in model organisms, and we talked about how most of it is pre and post spaceflight, which you mentioned. So, what kind of data would you need? And also, is there a pre-existing model out there that we could just fine tune, or would we need to train our own?
Yeah, that's a very good question. So the idea for this for this project is, I think when I look at the data portal, I think it's kind of includes everything we need. We just need to harmonize data and feed the data into our model. What we need is kind of the epigenetic features or gene expression, RNA-seq data across different conditions, like before the space flight, during the space flight, after space flight, something like that. And then we input use those kind of data as our output of the model. And in terms of the modeling, I don't think we need to train the model from scratch because there are many foundation models like Informa I just mentioned. It has been well trained based on the ENCODE data, some other consortium data. We can just actually somehow fine tune all of this pre-trained language models based on the space flight data, and in that way we can somehow kind of improve the modeling, the prediction performance, because the pre-trained model can already learn certain regulatory programs, regulatory mechanisms across different cell type tissues. So I think that's a kind of a straightforward strategy to for this project. Yeah.
That's great news that you don't think we have to train our own from scratch. Excellent. One more question. I see Wendy.
Oh, is somebody else asking a question or no?
I don't know. Evelina, I think you're unmuted. Wendy, go ahead with your question.
Okay, thank you. Great talk this morning. I'm very impressed with your algorithms. You're getting a lot of good things to help the neuroscience world.
Yeah,
I'm
thank you.
curious to see if you would be willing to, let's say in your searches, you've been looking at SNPs, and I'm wondering if you'd be willing to look at VNTRs. In addition, just given the Alzheimer's world is all over the place with the genetics, and we're finding a lot more associations with VNTRs lately. And so I'm wondering if your algorithm can take a look at those.
Yeah, that's a good question. So far we are kind of focused on the SNPs and also small indels in the analysis. But I think it will be very interesting to test the model to see if the model can also capture the impact of the structural variance, because I know that's also emerging field, because people realize the structural variance can also affect the disease risk and phenotypes. Yeah, but I think, yeah, definitely that's also a very good topic and direction in the future. Yeah. Yeah, thank you.
Thank you so much, Dr. Zhang. We do need to move on, but if you're able to stay for the rest of the meeting, maybe if folks have more questions, they can put in the chat.
Okay, awesome.
And I will be reaching out as we try to get this project off the ground.
Yeah, yeah, looking forward to it. Yeah, thank you.
Thank you so much. All right, Ignacio and Chris, who's going to be sharing the screen? We can make you a co-host.
I'll share the screen.
All right. All right, you should have screen sharing permissions now. So I am excited about our next talk from Ignacio Lopez-Francos and Christopher Teubert, which is titled Anomaly Response for Future Mars Missions.
Great. Yeah, thank you for this opportunity. So I'm Chris Teubert. I'm the portfolio lead for what's called Anomaly Response in the Mars Campaign Office. So to give a little bit of an introduction, right now for ISS operations, a lot of the operations relies on the ground. So this is especially true when things go wrong, when there's anomalies that happen. A lot of crew being able to respond effectively to that relies on calling the ground, which is fine for ISS where we're close to the Earth and communication is very quick. There's teams of experts on the ground that are ready to respond. For the most part, that's true for the Artemis missions, for as we get to the moon as well, with crewed operations to the moon, where we can rely on ground expertise to help support crew in responding to situations. But this whole way of approaching missions really breaks down when we start looking at Mars. So this was something that the Mars Campaign Office, when it started up, looked at. They were tasked with trying to enable a future crew's Mars mission. They looked at what are the main challenges that you need to solve to enable that Mars mission that you don't really need for current crewed space operations like that around ISS or even to the moon. And some of the things that they determined might be obvious. There's the effects of microgravity on the human body, radiation exposure, things like that. But one of the things that they found was this change of paradigm where crew has to be responsible for there's a set of problems that crew needs to be able to respond to independent from Earth that they don't have to in current operations. This is largely due to communication delays, which can be worst case scenario is around 20 minutes when Mars is the furthest one direction. So that's 40-minute round trip for communications. And this doesn't just mean that responses that have to happen in less than 40 minutes are impossible, but a lot of responses require iterative communications with the ground. And when you have a 40-minute comm delay integrated into each communication round trip, it really breaks down. And so they did a determination that there are a class of problems. The most significant of those is responding to faults that are where something breaks down that are unexpected. So there is not a predefined response that can be taken to this issue. And so for these, if the time to effect, the time to whatever critical effect of that fault is less than 24 hours, they found that really you can't rely on ground being part of that response. That crew needs to be able to respond to that class of problems independent from Earth. So that's what Anomaly Response, the portfolio that I lead, is about. So we're enabling this crew vehicle system to resolve these time-critical, unanticipated faults independent from Earth in a timely and effective manner. So as a project, we are developing technologies to help crew be able to do this. We chose two target systems to start with to develop these technologies. We're pretty far out from a crewed Mars mission. So right now, we are trying to develop technologies and techniques and guidance in order to enable that future crewed Mars mission. So we chose two different systems to use as our test systems for developing and evaluating these technologies. One is the urine processing assembly, and the other one is a power distribution system. So these were specifically chosen partially because they are so incredibly different. They're different in the way that they fail. They're different in the way that the signatures of the failure show up, and they're different in the responses that are possible for crew in each of these. Another difference that we chose is for the urine processing assembly, we're actually using data from ISS. We have 15 years of telemetry from ISS, including faults that have occurred on board ISS, and there have been many. And then for power systems, we are using data from simulation and testbed. And so this is important because with ISS data, we have real data to evaluate the performance of our approaches on real noisy, messy data. And it is very messy. Ignacio knows firsthand just how messy it is and how much work it is to prepare that data. But with power systems, we have clean data because it's from sim and a testbed, where we have absolute knowledge about what faults occurred and when, because we added the faults, and it's in a controlled environment where it's much less clean. So that allows us to prototype the approaches there in a cleaner and easier environment. So this is the architecture that we have to the work that we're doing so far, and I'm going to step through this bit by bit. But one of the things I want to take away from this is that there are a number of different technologies that are needed in order to allow crew to respond effectively to this class of anomalies. And these technologies are interdependent, so they rely on each other. It's my title is portfolio lead, but portfolio kind of implies that there's a bunch of different technologies that are developed kind of in stovepipes in isolation. That's not the case here. They're so interdependent and reliant on one another that it's really one combined anomaly response architecture in order to allow for it. I'm going to focus on mostly on the blue box for this talk because I think that's what this audience would be most interested in, since that's where we are most using AI and machine learning technologies. So starting with that. So the blue box here is fault diagnostics hypothesis generation. So essentially, the idea behind this is we are looking at sensor data. We're also looking at crew input, and we're trying to pull out information from those signals that might help crew respond effectively to a fault. So this can be, so I'm jumping to the bottom there. This can be things like, is there an anomaly that's occurred? Is there something unusual in the data? Can be isolating a fault that we know of, so that we know it's fault A or fault B, or even a list, a what's called an ambiguity set of different faults. So we it could be fault A, B, and C, hopefully with confidence numbers. Determining if the fault is not a known fault, so determining that's not one of the set of known faults that can happen. And this is important. This is something that happens actually quite often on crewed missions, even ISS, that's been up there for decades. There are faults that happen multiple times a year that just are completely unanticipated by the engineers that designed and prepared for the mission, just because not because of the poor performance of the engineers, but rather because we're human and there are limits to human imagination. And we're dealing with a very complex system of systems that are interconnected and interrelated. And there is no way to anticipate every way that that complex system can fail and every signature about what that failure would look like. So determining that it is one of these unanticipated fault modes, and then once you've determined that, pulling out other information that can help crew determine an effective response. So this can be things like localizing the fault to a specific component. So we can say it's an unknown fault. However, it's likely in component A. It can be identifying similar faults. It can be determining some trends and patterns that might be useful. So saying that it's an unknown fault, likely in component A, and it correlates with when this valve is changing state. That's something that crew might be able to work with. Or it's also part of the signature is that pressure is increasing, or even being able to identify a class of issues, like it's likely an electrical failure of some sort. So this is the kind of information that AR1 and 2 is trying to pull out to inform that effective response. As an approach, we're using all of the information available to us to build these models. So we're using expert knowledge when available. This is kind of the traditional approach to fault management with for crewed missions. So if and actually uncrewed missions as well, where you are encoding the knowledge that's in the FMEA, the knowledge of the experts that design the system that knows how things fail, into a diagnostic system. And mostly this is sensor thresholds. We're saying if pressure is above this, and if pressure is above this in pressure sensor A and pressure is below this lever in pressure sensor B, there's likely a leak or something like that. So we're encoding this kind of logic. So this is something that we use a tool called QSI Teams, not to be confused with the Microsoft Teams, to do this, and it's a diagnostic tool that will infer based on this expert knowledge. We also use physics when it's available. If we have a physics model of the system, you can utilize that with for diagnostics for these functions below. And I think what's of interest to this to this audience is we're also using the data. So a few more notes about the approach, and I'd like to I'd like to also mention that unfortunately in this presentation I don't have any of our initial results because that hasn't gone through the release process. But I'm going to talk through the approach and I'll give you some description of the results even if I can't show it to you. So our approach, constructive, we're building layers on top of a strong foundation. There's it's the toolbox approach. We kind of this was actually pretty important for management. Some of you that have pitched projects might be familiar with this phenomena where management might have an idea of a tool and say, well, why don't you use this tool? And the answer is yes, that tool can add something, but there with hard problems like this, it's never going to be one single tool or approach that is going to solve the whole thing. It's going to be that intelligent, thoughtful mixture of different approaches, each of which adding additional information that's going to bring you closest to the solution. So it's never going to be that some of the old era spaceflight people might say, just use QSI Teams. That's never going to give you the full picture. It's going to give you a picture for those anticipated faults because it's relying on the expert knowledge. Or some others might say, well, you throw a machine learning model on it. Well, okay, that works if you have the faults in the data that you use to train that classification algorithm. So there's advantages to each of them, and that thoughtful combination of those approaches is what we're taking. And also Crew is part of the solution. We're never going to be able to automate the whole problem. Crew has to be part of it. Some more technical considerations, kind of scope. So we have these complex systems of systems. The UPA the urine processing assembly is a complex system that includes a number of subsystems, some pumps and valves, and what's called the distillation assembly, which has been described to me as like a still in a washing machine, where it is essentially a still, but it has to be a rotating system. Because a still relies on gravity to do the separation. So it's a rotating system. And complex electrical systems and fluids storage and pipes. And so so one of the considerations that we had when creating this was the scope that you make the model. Do you make a model for the distillation assembly or do you make a model for the UPA? And the answer is, well, at this point, we mostly have a model for the UPA, but this hasn't been fully solved. We're considering architectures where we have actually smaller models that might be combined by some convolutional models that consider the effects between neighboring components or just fully connected layers to combine them to consider those effects. So that's one of our technical considerations as we're going forward that hasn't been fully resolved. Another one is timescale. So this is something that's interesting because the signatures of the faults show up in different timescales depending on the faults. So so I mentioned at the beginning the two systems that we're looking at, the UPA and electrical. And electrical is interesting because it shows the signatures of those faults show up in the high frequency data. So we're talking about 1,000 hertz data. When you can change from the time space to the frequency space, you can really see some of the signatures of these faults. Where a lot of the UPA faults show up in this kind of medium time scale, the measured in minutes rather than milliseconds. And then there's some faults that actually show up in these long scale time scales where they show up in days or weeks or months because they are slowly evolving degradations of the system. So our approach to that was actually that we are training different models on each time scale and combining the outputs of that. So as we bring in new data, it's separated and it goes through the models for each of the timescales. So I mentioned kind of the three sources of information. Um, as as we have different systems that are using these three sources of information, and also as we have the different models working on different scopes or different time scales, these are combined by what we're calling the recommender. And really, it's kind of a recommender is marketing, as you have to do in managing a project. It's an ensemble model approach, kind of for this audience. And where we intelligently combine the outputs of each of these, which are overlapping, but they aren't all providing the same output. So for example, QSI Teams is based on expert knowledge. It's able to only provide a diagnosis and anomaly detection for cases where the for cases that have been anticipated and pre-programmed rules have been defined by an expert as to how to detect these. So if it was a fault that showed up in the data after the design of the system, it's not going to be a part of that. And also, if these are more coarse tools, where they are limited based on the kind of simple rules that have been defined by an expert. A data-driven tool that's trained on instances of these faults may be able to create a better a better model for detecting some of these faults. And in the same way, a data-driven tool is going to be limited to faults that have been observed. And so there's advantages and weaknesses to each of these, and the recommender has to combine it intelligently. There's a few different approaches that we're taking to the recommender. The simplest is a voting-based approach, just kind of a simple combination, and that's more the baseline approach. And then we're looking at some neural networks to produce some sort of Bayesian estimate and combination of those. And that's more interesting because then you can train a neural network that will learn how much to weight the outputs in certain modes. So it may learn, in my example before, that the data driven performs really well at detecting this specific fault, and the team's QSI it performs decently well for the same fault, but maybe has some misdetections and some false positives because it's a simpler rule to define it. And so it will learn kind of how much to trust each of these and to combine in an intelligent way to come up with an assessment that's not just a scalar value, but is a probability distribution that's combined and used by future systems. All right. So machine learning pipeline, kind of, we're actually not that far into this project. It started in October. We had a little bit of work last year, but really in earnest, it started in October. But our general approach is an autoencoder, which does the anomaly detection. So I think this audience is probably familiar with an autoencoder, but I'll just say very briefly, it's trained on the nominal data to be able to reconstruct that nominal data as well as it can, so minimizing that reconstruction residual. And then when you put off nominal data in there, it will not be able to reconstruct as well because that's out of what it's been trained on. And that reconstruction residual is used to determine if there's an anomaly, and the magnitude of it is used to provide a score of that anomaly. So some sort of assessment of it, how confident. Confidence isn't quite the right word, so I'll keep a score. So some sort of score on whether or not there's an anomaly. We also train our classifier based on the reconstruction residual. We we want to so right now that's our approach. We want to consider training it not in the reconstruction residual, but on the latent space representation. So this encoded data, and evaluate the performance of the two. That's something that kind of talking to machine learning experts like yourself has been an advice that we've received, is that we may be able to pull out that signal a little bit more clearly from the encoded values of the data, the signatures of each of the faults. So this is a messy slide, but I'll talk to it. So basically, what I wanted to say. So this isn't a glitch. What I wanted to say here is that to the point I made earlier is this diagram just shows one autoencoder, but in reality, you have multiple ones working at different timescales of data to do the same assessment, and those are combined and used by the classifier. One thing we're considering is putting a mask between that, because once you get to hundreds and hundreds of sensors and you have multiple different time scales, you end up with quite a large number of values that are going into the classifier. So we're considering masks to pick out what are the sensors that are most critical for for identifying each of these faults, and then have separate classifiers for each fault and a mask that will only put through the values that are important for that fault to decrease the space, the input space there for the classifiers. So I know we're almost out of time, so I'm actually just going to say very quickly the other parts of it are being able to create an effective procedure for fault anomaly, for fault response, which these procedures are novel. So this is a real challenge, and being able to communicate that information effectively to crew. I guess any questions? This is what I had. Thoughts, feedback? I welcome feedback. We want to run this by as many people as possible and get feedback on our approach.
Thank you, Chris. Does anybody have any questions or feedback or thoughts for Chris? Yes, Ashish.
Hi, great. This is good work. Definitely for providing feedback, it would be useful to see some results that you are seeing, because I think
Yeah.
the procedure seems really good and comprehensive. But I think so what kind of anomalies that you're finding, or what extent the data are, and so on. So that would be useful to understand. But I think the methodology seems solid.
Okay. Good. So we are hoping to have our first paper here in the summer, after which we'll have some more of those kind of details that will be released to the public. And yeah, hopefully that will be an opportunity for that kind of feedback. But I'm happy to hear that you think the approach here is solid.
Yeah, so I work extensively on time series data from astronomy, various sky surveys, and including non-continuous ones. So it'll be quite interesting to compare some of the methods that we use for that. And
Yeah.
Hi, Chris. Brian Everts. This is really fascinating, and there's a lot to take in here. So I don't know that I have any like technical feedback, but just one thought. So working in this space, have you come across a company called Ontonics?
On tonics, I don't think I have.
They have an interesting software platform, and it may be interesting for some inspiration. So I think they're using some like entropy based approaches and like joint
Mm,
entropy and things to like multi-scale entropy to do some of the time series analysis, to do like some of the coarse graining and stuff. That might be something interesting to look into, to add on to this. But this is awesome work. It's again, it's a complex system. There's a lot to take in, and if something else pops into my mind, I'll definitely pass it on. But thank you so much.
okay. Thank you. I looked up on tonics here, so I'll read some more on that. Thank you.
I have one final question. Going back to your ML diagram and also keeping in mind the diagram that you have here where you have expert knowledge, physics, and data. So is this ML pipeline meant to fit kind of under data? Because it sounds like for the classifier, you kind of need to know the faults in advance. And you were saying for the faults that you can't really predict, those need to come with more expert knowledge or maybe physics-based knowledge.
Yeah, that's right. This is really zooming in on a majority of the existing data part of that pipeline. You can imagine separate to this, there's QSI teams working on expert knowledge and some physics-based analysis that then underneath would be combined by a recommender. And I have a diagram that shows that, but didn't go through the release process in time to include in this presentation, unfortunately.
Understood. Well, thank you so much. This was really fascinating, and we would love to have you come back again and give another talk since you're kind of looking for feedback as you continue building this out, since this is kind of new work. We also have an anomaly detection subgroup, but that lead isn't here today. So the next time that that you're able to come back, we could make sure that he's here and maybe have a dialogue.
Okay. Yeah, that sounds great. Thank you. And
All right.
thanks to Ignacio for helping set this up.
Thank you, Ignacio. Thank you, Chris, and thank you, Sai. We really appreciated your talks. Thank you for all the great questions and discussion. And we'll see everybody next month. Have a wonderful week.
Thank you, everyone.